Let's move on to the receptor tyrosine kinases, or the receptors with enzymatic activity. So, as with the G protein coupled receptors, this class of molecules binds ligand out here and conveys that event into the cytoplasm and eventually leads often to changes in gene expression in the nucleus. But unlike the G protein coupled receptors, there's not a single iconic structure associated with the receptor tyrosine kinases. There's definitely a theme, and that theme involves a relatively elaborate extracellular domain, a single transmembrane region, and then, of course, the tyrosine kinase activity that gives this group the name. It's a big family of proteins that don't quite share complete iconic structures with each other. And when the book makes a nice point in that single transmembrane domain is insufficient to couple a ligand binding event with a major conformational change in the cytoplasmic domain like you get with the G protein coupled receptor. So that these receptors have to do something different, and what they do different is shown on the next panel. But before getting to that, I wanted to just say a few things about these individual family members. The first is, here's the insulin receptor that we've actually kind of talked about. It is a receptor tyrosine kinase, but it's different enough from these in terms of its structure and the way it behaves that I'm going to postpone it until later on. The mother of all receptor tyrosine kinase is this one, the epidermal growth factor receptor. So a lot of what I say is going to be really about it, but it applies to these other members as well. Nerve growth factor receptors we're talking about because of something that I mentioned earlier, and that was that signaling endosome idea. So if you realize what has to happen with a nerve growth factor receptor, it's going to be, in many cases, at the end of a very long axon, and what it needs to do is convey information to the nucleus, and the nucleus is going to be at the other end of that cell. So here's that receptor. So you can probably get the point that being on an endosome would help a lot, because you just bring that endosome down here so that it can then signal to the nucleus, and that is exactly what goes on. Another point I want to make is just a personal one, and that is this VEGF receptor. This is one that is particularly significant to me. It's a fairly standard receptor tyrosine kinase. VEGF is the growth factor. In the case of macular degeneration, or at least age-related macular degeneration, it is a receptor that goes haywire and leading to the disease, and that disease is treated in many cases with antibodies to VEGF to block signaling from this pathway. And the reason I know that is because I have macular degeneration and I have that treatment myself, and I'm very appreciative of the people that did the work to figure that out, because the treatments work pretty well. But now let's get to the bottom of the signaling pathway, how it is that ligand binding here can result in signaling inside the cell. And the way that's shown in schematic form is on this figure 1544 from your text. So as I've mentioned many, many times now already, the inactive form of most receptor tyrosine kinases is monomers. The insulin receptor is an exception, but most of them are monomers. And one of the things accomplished by the growth factor as a ligand is the bringing those monomers together. And it's very often the case that it is the proximity of two homodimers that leads to the activation of an autocatalytic activity that often involves phosphorylating one of the monomers by the other monomer and vice versa. So that as a result of that dimerization, in some cases trimerization, you get ultimately the formation of an array of phosphotyrosines along the cytoplasmic surface, in some cases a lot of them along extended regions of the receptors that serve as the beacons for the signaling. Now, maybe counterintuitively, what doesn't happen is that these tyrosine kinases don't usually phosphorylate something else that then goes on to do the signal transduction. That actually kind of happens with the insulin receptor, but it's unusual in that regard. What usually happens is that those phosphotyrosines serve as recruitment signals that bring in other proteins that do the work of the signal transduction. And those proteins are shown sort of schematically in this diagram, with the appropriate indication that you can have from a single phosphorylated receptor tyrosine kinase multiple effector molecules binding to multiple phosphotyrosines and then triggering what in some cases are very, very complicated cross-talking signals into the cytoplasm, leading to major developmental changes in the cell that undergoes it. Now, we'll talk specifically about those pathways, or at least two of them, but while I'm here, I'll just go ahead and tell you that we're going to simplify things by just indicating that among the important downstream pathways associated with this phenomenon, this activated tyrosine kinase receptor in the phosphotyrosines, are going to be the so-called MAP kinase pathway and the PI3 kinase pathway. And because these pathways are associated with major phenomena like growth, multiplication, proliferation, whatever you want to call it, and survival, roughly broken down in that way, but of course there are exceptions. These are very important phenomena associated with things like development and just the normal functioning of a multicellular organism, and they're often the things that very often go wrong in the case of cancer. So these things are very, very actively researched. So before going into those specific pathways, let me just give you a couple of other examples of how that's going to work with particular receptors singled out. So here's specifics of the activation of the EGF receptor. I don't care that you know exactly the mechanism of the activation of the tyrosine kinase, but I do want you to appreciate this length, and that it's actually oversimplified here. There are in fact 7 phosphotyrosines in the cytoplasmic tail of the EGF receptor that recruit different proteins whose domains allow them to recognize phosphotyrosines. And I'm only going to have time to talk about a few of these. We actually know this one. We know phospholipase C anyway. This is like the gamma variety of that enzymatic activity, but it does what phospholipase Cs do, and therefore leads to those calcium spikes and protein kinase C activation. We're going to focus on this one, GRAB2. It's going to be the next thing that I talk about specifically. But then if you look at the platelet-derived growth factor, another very well-researched receptor tyrosine kinase, it's kind of unusual in having a split tyrosine kinase domain. 1, 2, 3, 4, 5 different phosphotyrosines that recruit and activate different pathways. Here's the other major one that we're going to be talking about, the PI3 kinase pathway. But there's the phospholipase C gamma again, and one that we're not going to talk about. Never mind these domains just yet. We'll get to those in just a second. So in talking about those SH2s and SH3s, I think it helps to actually work backwards. So if we're going to focus for the time being on the MAP kinase pathway, then the function ultimately of the phosphotyrosines is to activate a small g protein that I'm sure you've heard of before, kind of the mother of all small g proteins, called RAS. So RAS is the name of the family that includes most of the other G proteins that we've talked about, not the heterotrimeric ones, although it does have a RAS domain, but things like RAB and ARF, those are members of the RAS family. But in our case, we're actually going to use RAS itself. So this is a small G protein that is anchored to the membrane by a short fatty acid anchor. And as all good G-switch proteins, it is active in the GTP bound state and inactive in the GDP bound state. It pretty much stays on the membrane where it's in the same plane as the receptor that ultimately is going to be in charge of controlling it. But the way that works is through, as with all good G-switch proteins, its activation by a GEF. In this case, the GEF that was first discovered in the context of receptor tyrosine kinases, something that's referred to as SOS, which is short for son of sevenless. And don't get too carried away in terms of the 7. This is not going to be a G protein coupled receptor 7. This is a particular phenotype associated with Drosophila, and the terminology stems from Drosophila terminology, which is always kind of clever. But the 7-less is actually a phenotype mutation in a protein called 7-less that is very, very closely related to the mammalian EGF, abnormal growth factor receptor. And it's called 7-less because the wild-type pigments in the Drosophila eye have 7 of these things, whereas the mutant has less than 7. So here you have 6 of those that are 7 in the wild type. So that's where the gene name 7-less came from. Son of 7-less was discovered in a screen of Drosophila mutants and has a fairly similar phenotype to 7-less, so hence the name. It's referred to in the pathway as SOS, and it's going to be the GEF that activates RAS. It itself is recruited to the membrane where it can encounter RAS in the inactive form and catalyze that guanine nucleotide exchange by a protein that serves as a link between the activated receptor and its phosphotyrosines and the guanine nucleotide exchange factor. That protein was actually discovered in yeast, and that's why it has a relatively boring name, either GRB2 or GRAB2. But its role is to serve as the link between the activated receptor and ultimately the activation of RAS. It does that through these domains that I want to talk about specifically in just a second. But for now, I'll point out that the phosphotyrosine is recognized by one domain on this protein, the so-called Sark homology group 2 domain, whereas SOS is recruited by virtue of the 2 SH3 domains on GRAB2. The overall pathway is growth factor bonds triggers the dimerization and autophosphorylation of the cytoplasmic domain, which then recruits the SH2 domain of GRAB2, which by virtue of having 2 SH3 domains recruits SOS, which then activates RAS, and that ultimately leads to the MAP kinase pathway that we'll talk about in just a minute. Before doing that, I want to complete this video by saying a little bit more about these SH domains. So once again, S is Sark, and the Sark story is a great one if you have time to read up on it. I'm not going to go into what Sark is specifically, and we're not going to need it very much for the rest of the semester, but it is a very important tyrosine kinase in its own right. But its main claim to fame these days is by having these domains in them. And these domains are actually independently folding in the traditional cell physiology exam 1 sense. The SH2 domain is a relatively small domain that can be plugged into just about any protein without affecting anything else that protein does. And that's why it's so common as a way of making a protein recognize phosphotyrosines. There are in fact other phosphotyrosine recognizing domains One is the so-called phosphotyrosine binding PTB domain, but it's not nearly as common as SH2, and it's just not quite as useful when it comes right down to it. But the SH2 domain is very useful and can be plugged in, and you see this diagram. It's in your text, but you see it elsewhere, where the way it works is that you have the recognition of phosphotyrosine and then some other context recognizing domain, and you can just stick that into any protein, and all of a sudden you have something that will now serve as a protein that can be regulated by tyrosine kinases. So here's SARC with it has an SH1 domain that is not particularly famous anymore. SH2, SH3, and then the tyrosine kinase of SARC. GRAV2 is essentially only those domains. It's a relatively small protein that has a single SH2 domain like so, and then two SH3 domains. So that's all it, no catalytic activity. All it does is bind to phosphotyrosines and then recruit those proteins that recognize SH3 domains. And usually that recognition is through proline-rich regions. So that, for example, SOS has proline-rich regions that recognize, or proline-rich domains that recognize the SH3 domain, so GRAV2, and that ultimately leads to the activation of RAS. So talked about phospholipase C gamma as being involved in or activated by receptor tyrosine kinases. It has both SH2 and SH3 domains along with the catalytic domain, and then we'll eventually get to the PI3 kinase pathway as one of the two main things that happen when receptor tyrosine kinases get activated. One of the two subunits of PI3 kinase has SH3 and SH2 domains, in addition to the binding domain for the other component of the PI3 kinase. We'll talk about those specifically in the next video.